So today we're going to start an experiment on bacterial conjugation. We have a donor that's going to contribute a plasmid that codes for tetracycline resistance to a recipient that has a resistance gene for kanamycin on the chromosome. So the first thing I'm going to do is mix the donor and recipient. The donor to recipient ratio is usually 1 to 10 so that every donor encounters a recipient. So with that in mind, let's mix the donor and recipient. We'll put it in the 37-degree water bath. Bacteria are happily mating. All right. So the bacteria have been mating for a minute. I'm going to add a little more tryptic soy broth. Put it back in the water bath. And then in 4 minutes, we'll sample this mating mixture. So while the bacteria are mating, I'm going to inoculate the controls to make sure they are what we think they are phenotypically. So when you're picking up organisms from a slant, you have to wait 8 seconds for that loop to cool. So this is BB4, the donor, single street. Single street. We're just looking for growth to make sure that BB4 grows on the tetracycline plate and 2507 grows on the kanamycin plate. We'll sample 100 microliters from our mating mixture. Spread plate it on the kanamycin and tetracycline plate. So, after mating, what would we expect? If there was transfer of the tetracycline resistance plasmid, we would expect a lot more colonies if conjugation worked on this plate, right? So if the conjugation experiment worked as planned, we should expect on that 30-minute plate a lot more colonies that show there was resistance, transfer of the resistance plasmid, the tetracycline resistance plasmid from donor to recipient. So that increase in number of colonies will tell us that the experiment worked. Okay, we're going to look at our results of the conjugation experiment. Remember that we mixed a donor and recipient. The donor coded for tetracycline resistance, the recipient coded for kanamycin resistance. The donor contributed a plasmid to the recipient, and we sampled at 5 minutes and 30 minutes from that conjugation mixture. And let's look at the results, and if conjugation worked, we should expect to see an increase in the number of transformants, or I should say transconjugates, that are resistant to both kanamycin and tetracycline. So let's look at the plates. Okay, at 5 minutes, we can see individual colonies, 482 colonies. All right. At 30 minutes, if the conjugation experiment worked, you would expect to see many more colonies, and there are probably millions of colonies. You notice that the growth is a complete film. So this is a lot more growth than this plate. it shows that the conjugation worked much better than actually I expected. Now, let's step back. What about the controls? Are they behaving the way they are supposed to? So we would expect that BB4 would grow only on the tetracycline plate and not on the kanamycin plate. Conversely, 2507, resistant to kanamycin, grows on the kanamycin plate. And we have a little bit of faint growth that I don't like on the tetracycline plate, but you get the idea.